Today is June 28th, 2024. My name is Casey Norman, and today we're going to be doing a blood cross match. Normally you would do these prior to a blood transfusion between the recipient and the donor. These are performed to see if there's going to be any sort of serological incompatibility between the two samples, to avoid the recipient experiencing a severe reaction. So my samples here. I'm going to start by taking my 2 EDTA tubes with the sample, and I'm going to centrifuge them at 9,000 RPM for about 5 minutes. Okay, so once I have spun down my 2 purple tops, I'm going to take a pipette and my donor Dixie. I'm going to take the plasma very carefully by pipetting it out and placing it into clear tube. Close these up, and I'm going to do my reset. Back on, and so now I have my plasma for my donor and my reset. Next, you're going to do a rinse on your red blood cells. So what we're going to do is we are going to add some sterile saline to our red blood cells in the tube. And we are going to spin them again. Put these back in the centrifuge. We're going to do this a total of 3 times. So for your red blood cell rinse, you will spin them down, separate, and pull off the top layer and discard. We'll do this a total of 3 times. So I'm going to remove the liquid part and then add some more saline and centrifuge again. So 3 times on the red blood cell rinse. Okay, so after I've completed 3 of the red blood cell rinses, I pull the excess off the last round, and I have the red blood cells left in the purple top. I've pre-made a clean tube with 20 drops of saline, sterile saline. So I will add 1 drop of the red blood cell to my 20 drops of sterile saline. So for my donor, I'm going to take a pipette and get 1 drop place it in. This is going to be my red blood cell solution, or suspension, better yet. So for my recip, take 1 drop. Of the red blood cells. And now I have my recip red blood cell solution, suspension. So now that I have my plasma and my saline, or my red blood cell suspension, I'm going to take my control tube and I'm going to put 2 drops of the recipient plasma, 2 drops of recipient plasma, 1, 2. And I'm going to add in 1 drop of my recipient red blood cell suspension. Okay. For my minor, you're going to take 2 drops of the donor plasma and 1 drop of the recipient red blood cell solution. So 2 drops of donor plasma and 1 drop of my recipient's red blood cell solution. For my major, I'm going to take 2 drops of the recipient plasma, mix it with 1 drop of the donor red blood cell. Now, I'm going to take these and gently mix them and allow them to sit for 15 minutes in room temperature. And then I'm going to centrifuge them for 15 seconds, and then we will check for agglutination. So once we have allowed our control, minor, and major to sit for 15 minutes, we centrifuge them and for 15 seconds, and then you're going to take them out, and there's going to be a little pellet at the end, or at the bottom. So you just tap on it a couple times, and as it resuspends, you can kind of see. So this is my control, and you wouldn't see any reaction or agglutination. So once you've resuspended your red blood cells, you will take a drop, take a little sample from each and dispense it onto the appropriate labeled slide. So I have my control. Here I have my minor, so I'm just gonna tap the little pellet until it is resuspended. I'm gonna take a clean pipette, take 1 sample, or 1 drop from the sample, place it on the appropriate labeled slide. So over the next couple of minutes, we're gonna observe these slides, and I'll get my camera. So we'll observe these slides over the next couple of minutes for any reaction between the recipient and donor samples. You will see kind of an agglutination. I don't know if you can. So for a canine's first blood transfusion, you don't necessarily have to do a cross match, but anything after any other transfusions after about a week, you will definitely want to do this to avoid any significant reaction to the donor or between the donor and the recipient's blood. So we'll come back in a couple of minutes and check these samples. So after about 2 minutes, you can see that there's no significant agglutination of either of the samples. So this would be interpreted as it would be okay to go ahead with the blood transfusion between this donor and this specific recipient.